Hi, hello, I'm Dr. Fishman, and I would like to brief you today about Can-Fite BioPharma, the company profile, and our activities. The next slide is our forward-looking statement, and let's move to the next one, which presents the company profile. The company has proprietary core technology where we are developing small molecule orally bioavailable drugs which target a specific cell surface receptor. We are looking at anti-inflammatory, anti-cancer, and anti-metabolic effects of our drugs. Financially, the company is dually traded on the Tel Aviv and on the New York Stock Exchange. Due to end of Q2, we had $8.2 million in the bank. We have 4 million ADRs outstanding and 120 million ordinary shares. We operate both from Tel Aviv in Israel and also from the States. In Tel Aviv, we have the Discovery Labs and the company management, whereas in the US, we have the preclinical and clinical operation. We have leading key opinion leaders on our clinical advisory board and successful corporate partnerships, which I will brief you about it shortly. We have initiated our program based on a very interesting clinical observation. The cancer very rarely metastasizes into striated muscles. What we found was that muscle cells can produce and release small molecules which have robust anti-cancer and anti-inflammatory effect. This molecule induced apoptosis of cancer and inflammatory cells, and what we did was to mimic the muscle-derived molecules to make them synthetic, and these are the molecules in our pipeline today. What we know also today is that we have a therapeutic target, the A3 adenosine receptor, very highly expressed on pathological, inflammatory, and tumor cells, but not on normal body cells. The drug products are small molecule, orally bioavailable. We have proof of concept from phase 2 clinical studies, which I will share with you the data. And last but not least, the target is also serves as a biological predictive marker. So in our pipeline today, we have piclidenoson, which is currently positioned for rheumatoid arthritis, phase 3, and also for psoriasis, phase 3 clinical studies. Rheumatoid arthritis targets $35 billion markets, and the psoriasis $11.48 billion markets. Namodenoson is positioned for liver indications due to the fact that the drug is extremely stable. So we are treating liver cancer, a $3.8 billion market, and NAFLD/NASH, which are $35 billion markets. Not only that, but we recently signed a partnerships with Univo Pharmaceutical, and we are also targeting the development of cannabinoid-based pharmaceuticals. So next slide, in the frame of this partnership with a cannabis company in Israel, Univo Pharmaceuticals, we are developing new cannabinoids for pharmaceutical purposes and also for A3 adenosine receptor-based assays. All of it is based on the fact that cannabinoids are inducing their effect not only through CB1 and CB2 receptors, but also by targeting our A3 adenosine receptor, and by looking at the cannabinoids and see how they bind to our receptor, we can look at new cannabinoids and new clinical indication based on our experience with the target and with the indications which I have already mentioned. We are looking here at a huge market of $56.7 billion by 2026, and we do hope to be positioned nicely on this market. In the next slide, I would like to share with you the corporate partnerships that we have already signed. Cipher Pharmaceuticals, a Canadian company, Gebro Pharma, a European-based company, CMS, a Chinese company, and 3 Korean companies Kwangdong CKD and Kyongbu, with whom we signed agreements which entail out-license of both piclidenoson and namodenoson. Overall, we have received $18 billion as upfront money and more milestones, regulatory milestones, sale milestones, and two-digit royalties will come down the road out of these out-licenses. So our first drug candidate is piclidenoson. It's a small molecule, orally bioavailable drug, a small one. It is an adenosine derivative. It has a definitive mechanism of action where it targets two signal transduction pathways in the cells, the Wnt and the NF-kappa B, giving rise to apoptosis of inflammatory cells. Today, piclidenoson is positioned for rheumatoid arthritis and for psoriasis. Just to give you a flavor of the data of our phase 2 clinical study in rheumatoid arthritis, we reached the primary endpoint, which was ACR20, and the drug did a very linear job. On the right part of the slide, you can see a subpopulation who hasn't been treated before with any DMARD, and basically our drug is first line, and the response was very robust, similar to that of the biological drugs on the market. Currently, and please look at the next slide, in the frame of the phase 3 clinical study, we are treating only this patient population of those who haven't been treated before. Our drug is first line. We have 4 arms, piclidenoson, 1 mg, 2 mg, a comparator methotrexate, and placebo. We are currently enrolling patients and hope to next year to be done with the enrollment and very early 21 to come up with the data. The next indication is psoriasis. We are also conducting a phase 3 clinical studies. Interestingly, during the phase 2-3 clinical study, we haven't reached the primary endpoint since it was on week 12 and the drug didn't show any difference on week 12. However, when we look down the road, we can see a very nice effect of our drug when we are comparing it to Otezla, the Celgene drug, which has been launched a couple of years ago. And you can see the Otezla is the pink line and our drug is the blue line. And you can see a superiority of our drug versus Otezla while we are looking at PASI 50, PASI 75, PASI 90, and PASI 100. So currently we are conducting a clinical study, again 4 arms, 2 mg, 3 mg, and a comparator Otezla and placebo. We are actively enrolling patients, and the timelines are similar to those of the rheumatoid arthritis. In the next slide, I would like to present you with our second drug candidate, namodenoson. It's a very stable drug, similar in its chemical entity to the other one, very similar mechanism of action. And we decided to position it for liver indications due to the stability of the molecule. And today it is positioned for advanced liver cancer and for NASH. As of the liver cancer, we have recently concluded the phase 2 clinical study, and we just concluded a very successful end of phase 2 meeting with the FDA, where we have presented our data and also presented the phase 3 protocol, which we have agreed with the FDA on the phase 3 protocol, and we are going to initiate the phase 3 protocol next year. Just to summarize the regulatory status of the liver cancer, we have been granted by the FDA and by the EMA an orphan drug status, a fast track status by the FDA. End of phase 2 meeting was successful. We have concluded it 2 weeks ago with the FDA and agreed on the protocol. Last but not least, we have a compassionate use program in Israel where we are treating advanced liver cancer patients who do not have any other drug on the market. I have already briefed you about the phase 3 design, which we envision to initiate next year. Last but not least, I would like to share with you the indication of the NASH. The NASH is a huge indication today with no registered drug yet on the market. We went through very excellent preclinical data and a couple of experimental animal models where we have concluded anti-inflammatory, anti-fibrotic effect, anti-steatotic. We have decreased statistically significantly ALT and also induced a liver protective effect. Altogether brought us to the conclusion that we should go ahead and initiate a phase 2 clinical study where you can see the design here. We are comparing namodenoson in 2 dosages to a placebo. We concluded patient enrollment quite recently, and we are going to come up with the data and release the data Q1 2020. Just to conclude, I would like to share with you a spotlight on our milestones. So the NAFLD-NASH phase 2 data will be on Q1 2020. The liver cancer phase 3 preparatory work is currently done, and we are going to initiate it H1 2020. Piclidenoson enrolls patients for 2 phase 3 clinical studies, rheumatoid arthritis and psoriasis. Thank you very much and for your attention, and I will be very happy to answer questions. Thank you.
Okay, we're now going to open up our line for the Q&A session. If you do have any questions for Dr. Fishman, please just click the question button below the presentation window, and you can type your question in. We'll get to those in the order that they do come in. With that, we'll just jump right into some of the first questions here. Dr. Fishman, our first question, with your new relationship with Unovo, you mentioned working to create CBD and A3AR assays, but could you talk a little bit about how the company plans to monetize these assays and how the relationship is set up for advancement of any candidates into IND-enabling studies that you discover through these assays?
Of course, thank you. The first steps are to select the right drug candidates based on in vitro and in vivo assays that will be conducted at the Unovo labs based on Can-Fite knowledge and expertise. Upon selection of the most active candidates, the top companies will share the expenses and later on the profits, and this will take place down the road, of course. So we are ready to go, and we are very shortly initiating our collaborative work with Unovo.
Very good. Thank you, Dr. Fishman. And our next question, can you talk about what would be considered a win for the Acrobat trial? Is it just beating the placebo and showing non-inferiority to MTX with perhaps better safety, or do you need to show superiority to MTX to really feel like you have a drug there?
Sure. So based on the protocol, we need indeed to show non-inferiority versus methotrexate. However, based on the data of the former study in the subpopulation, which included naive patients, and is a population that we are currently including in our study, we are hoping that the data will be reproducible and will be superiority as per methotrexate.
Thank you, Dr. Fishman. And our next question, can you talk a little bit about the current cash balance, the expected burn rate, and any financing plans?
Yes, actually, we are going to come up with our Q financials very shortly during the next couple of weeks after the board meeting that we are going to have. Due to June 30, we had $8 million in the bank. You may know that via our collaborative agreements that we have already signed, we will get more money when our phase 3 liver cancer study will be initiated. And then more options are on the table, of course, and we will share it with the public down the road.
Very good. Thank you, Dr. Fishman. This is back to the Acrobat. Are you pre-screening patients for A3AR, or is that just going to be a subset analysis? And if it's just a subset analysis, will you have those data at top line, or will we have to wait to see those data?
Sure. So the A3 adenosine receptor is going to be a subset analysis, and based on the data of the present study, we will be able to conclude it if it is a companion biomarker or not at the end of this study. It will be released in the top-line results or full data sets as we will be able to run the A3 adenosine receptor bioassays.
Thank you, Dr. Fishman. Our next question, can you talk about what would be considered a win for the COMFORT trial? Otezla is obviously a very successful drug, so is your goal to show similar initial response with better durability or on the ACR, or do you think you have to show superior ACR numbers to Otezla to have a real market opportunity there?
Based on the analysis of the former phase 2-3 study and the post-hoc comparison to Otezla, the response to the latter is faster. However, it plateaued after 12 weeks, whereas piclidenoson has a very linear way of action. So to understand the full effect in the current study, we are treating the patients for 48 weeks. We also need to keep in mind that we increase the dose, therefore we could have even better results.
Thank you, Dr. Fishman. And our next question, what work is ongoing with evaluating A3AR as a biomarker, and how are you incorporating it in ongoing clinical studies?
Yes, actually, the company has developed a bioassay to look at the biomarker, and the assay is conducted in a central lab where all the blood samples of patients from all our clinical studies are analyzed. We do believe that it can be a companion biomarker, and when we will conclude the psoriasis and the rheumatoid arthritis studies, we will be more knowledgeable and be able to come up and claim if it can be a companion biomarker.
Very good, thank you. I'm not sure this is a full question here, but somebody's asking about patient enrollment in a trial for colon cancer. Can you provide any information around that?
Actually, we are not conducting a colon cancer. We have conducted a hepatocellular liver cancer study where we have, of course, completed enrollment, came up with the data recently, and also approached the FDA with the phase 3 protocol that has been blessed by the FDA. It was quite recently, 2 or 3 weeks earlier.
Very good. Thank you. And our next question, for the P3HCC study that you plan to start next year, assuming you hit in that trial, is that enough to file for approval with the FDA, or do you think you would need to do another confirmatory study in HCC?
We expect, based on the FDA meeting, the end of phase 2 meeting that we have conducted recently, we expect that one trial will be enough to register the drug with the FDA.
Very good, thank you. And another question on the P3HCC. What are your thoughts on financing that particular trial?
Yes, upon initiation of the HCC phase 3 clinical trial, it will trigger milestone payments under some of our licensing agreements, and we are looking to partner the drug for this indication as well.
Very good, thank you. And our next question, several big pharma players in NASH are pursuing combination therapy with their NASH assets. The Gilead P2 Atlas study comes to mind. Do you think about the potential for combination studies with Nemodinesen? And is there a mechanism of action in NASH that you think might synergize really well with the drug for NASH patients?
Indeed, yes. The Gilead Atlas indeed combined two drugs. Both affect steatosis. We do know that our mechanism of action includes anti-steatotic, anti-fibrogenic, and anti-inflammatory effects. At this point, we are waiting for the top-line data of our phase 2 study, NASH study, which is scheduled to be released during Q1 2020, and the data will guide us whether to continue the development as a standalone or in combination.
Very good. Thank you. And that looks like that might have been our last question that's come in today. I guess just in summary, there's obviously a lot of detail in the presentation, and especially here in the Q&A that's been provided, but just sort of in summary, what's the key takeaway? I mean, why should investors take interest in Can-Fite today?
Sure, thank you. We think that we have drug candidates in advanced stages of development. The profile of the drugs, all our drugs are small molecule, orally bioavailable with excellent safety profile. We know it since we have already dosed more than 1,000 patients with our drug. So, and the mechanism of action is also unique. And last but not least, we target only the disease, the pathological cells in the body, and the normal body cells are refractory to the effect of our drug. We think that our drugs has a great potential, and that the deals that we have already signed till today just point out that we can come up with more deals. And these are the reasons that we think that we can position ourselves nicely with the drugs that we have. And of course, we think that this was good for investors to hear our pitch. And of course, we will continue coming up with more PRs and make the investors part of our company. Thank you.
Very good, and thank you, Dr. Fishman, for sharing the story. A lot of exciting things going on with the company. For our viewers, obviously there'll be a lot of announcements on progress with these various initiatives. So if you want to stay abreast of that, make sure you sign up for our free news alerts. You can do that right here on the page, and then that way you're one of the first to get the news as it does come out. And with that, again, Dr. Fishman, I want to thank you for sharing the story today, and I want to thank our viewers for participating.
Thank you.